Welcome to Lecture 11.4, in which we will go over proteins and translation. This is Dr. André Loff, who received the Nobel Prize in 1965 along with François Jacob and Jacques Monod for discovering the relationship between genes, enzymes, and viruses. This is Dr. Marguerite Loff, André's wife, who worked and published many papers with him, but unfortunately did not receive as much recognition as her husband. Dr. Har Gobind Khorana was the first to synthesize genes and use these techniques to show that peptides can be made using synthetic mRNA as templates. For this work, he received the Nobel Prize along with Dr. Robert Hawley and Dr. Marshall Nirenberg. Our learning objectives for this video are to explain the role of translation in the central dogma of molecular biology, define tRNAs and rRNAs and their roles in translation, understand the role of codons in translation, describe the main steps of protein synthesis, and understand the directionality of proteins. Reviewing the central dogma of molecular biology again, we have now talked about the process of how DNA is used to make a complementary strand of mRNA. We will now go over how the mRNA is used to make proteins. Recall from the macromolecule lecture that the building block of proteins are amino acids, which determine the protein's shape, and the shape of the protein in turn determines its function. Eukaryotes generally use 20 different amino acids. Amino acids share the same basic structure and are named for containing an amine group and a carboxylic acid group. Each amino acid has a different side chain, and these side chains determine the property of the amino acid. Translation is the process of using the mRNA transcript to obtain the amino acid sequence of a protein. In other words, the nucleic acids are translated into amino acids. This process is performed by ribosomes, which are the protein-making machines found in the cytoplasm and embedded on the rough ER. The mRNA sequence is translated 3 nucleotides at a time, and is read in the 5' to 3' direction. The set of 3 nucleotides are called codons, and each codon corresponds to either an amino acid or a stop codon. Like replication and transcription, translation can be divided up into the stages of initiation, elongation, and termination. In the previous video, we introduced that tRNAs and rRNAs are used for translation. We will now talk a bit more about their roles. Here is the structure of a tRNA. Although the RNA is single-stranded, it folds into itself to create the shape seen here, with various base pairs within the same strand forming hydrogen bonds with each other. In purple are the nucleotides that make up the amino acid binding site. In gray are the 3 nucleotides that make up the anticodon, which is a set of 3 nucleotides that are complementary to a codon. This allows the tRNA to recognize a specific codon, and it will bring the amino acid that corresponds to the codon during translation. Although the tRNA is called the transfer RNA, in a way it is also the translation RNA, as it is essentially the translator between the mRNA and the amino acid. Here is a simplified tRNA structure. This is the 5' end, and the amino acid binds to the 3' end. The anticodon binds to the codon on the mRNA. As with all other instances of 2 nucleic acid strands interacting, the anticodon is both complementary and antiparallel to the codon on the mRNA. The ribosome facilitates translation and allows for the formation of proteins. It is itself composed of multiple ribosomal proteins and rRNAs. While the protein portion of the ribosome was once thought to be the part that actually carries out translation, it is now known that the rRNAs are the ones that catalyze protein synthesis. rRNAs also make up the majority of the RNA found in cells. The ribosome as a whole can be divided up into a small subunit and a large subunit. The small subunit is responsible for binding to the mRNA to begin translation. The large subunit binds to the tRNA and links the amino acids together. It has 3 tRNA binding sites. The A site is essentially the amino acid site, and is where the tRNA enters the ribosome with an amino acid, with the exception of the first tRNA. The P site is where the tRNA with the growing polypeptide chain moves to. It is also where the first tRNA binds. The E site is where the tRNA goes before exiting the ribosome. Codons are essentially the codes that are used for translating nucleic acid sequences into an amino acid sequence. Since there are 4 possible nucleotides in mRNA, and they are read in triplets, the total number of possible codons is 4 to the power of 3, which is 64 codons. Of course, this is more than enough codons to represent the 20 essential amino acids, as well as stop codons. This brings up the concept of redundancy, also referred to as degeneracy, which is that many amino acids and the stop codons are represented by multiple codons, although the number of possible codons for each amino acid can vary. Here is a codon wheel. To use this wheel, we start at the center, which is the 5' end of the codon in the mRNA. To figure out the amino acid that corresponds to a codon, we start in the center of the wheel, then work our way towards the outside of the wheel. For example, AUG is what makes methionine, the start codon. Methionine and tryptophan are only represented by one codon, while a few other amino acids, such as leucine, are represented by 6 codons. The redundancy is helpful because if, for example, UUG becomes mutated into UUA, the resulting amino acid is still leucine and there is no effect on the protein sequence. We will continue to use the codon wheel as we go through the process of translation. The initiation step of translation begins with the ribosome binding to the 5' cap of the mRNA and looking for the start codon, which the first tRNA will bind to. Initiation itself can be broken down into 3 steps. In step 1, the small subunit of the ribosome binds to the 5' cap of the mRNA. In step 2, the small ribosomal subunit starts scanning the mRNA in the 5' to 3' direction for the start codon. Remember that the start codon is AUG, which is also bolded in the mRNA here. Once the ribosome scans and finds the start codon, the tRNA with the start codon, methionine, recognizes and binds to it. Since the diagram shows the 5' to 3' direction of the mRNA from left to right, the tRNA and the anticodon, CAU, must be in the opposite direction. The tRNA then binds to the P site of the large ribosomal subunit. Now that the key players have been assembled, elongation can occur. Amino acids will start being connected together to form a polypeptide chain. Elongation can also be broken down into 3 steps. In step 1, the tRNA with the next amino acid enters the A site. Since the codon is GUC, the next amino acid is valine. In step 2, the amino acid in the P site is bonded to the amino acid in the A site. The tRNAs will then move to the next site, with the tRNA that was in the P site moving to the E site, and the tRNA in the A site moving to the P site along with the growing polypeptide chain. This process then continues with the rest of the mRNA. The next codon is GAG, which codes for glutamate. The tRNA enters the A site, and now the valine in the polypeptide chain bonds with the glutamate. Once again, the tRNAs move to the next site over, and so on. Eventually, a stop codon will appear. In this case, the stop codon is UAG. Once the stop codon appears in the A site, the ribosome will recognize it and proceed to dissociate or break apart. This releases the polypeptide chain, while the ribosome subunits split apart. Let's quickly review how peptide bonds are formed between amino acids. Since amino acids always have an amine group and a carboxylic acid group, they also have directionality like nucleic acids, with the amine side referred to as N for the nitrogen, and the carboxylic acid side referred to as C for the carbon. Like the formation of many other polymers, peptides are made through dehydration synthesis. The carbon on the carboxylic acid of one amino acid forms a covalent bond with the nitrogen on the amine group of the second amino acid. In other words, the C end of one amino acid joins with the N end of another amino acid. The C end of that second amino acid can then go on to form a bond with another amino acid. The first amino acid is always at what is called the N terminus, while the end of the polypeptide chain is referred to as the C terminus. In this video, we learned that translation is the process of using mRNA to make proteins. The tRNA is responsible for recognizing the mRNA and bringing amino acids to the ribosome to form a polypeptide chain, while the rRNA is a part of the ribosome that facilitates translation. Codons are a set of 3 nucleotides on the mRNA that correspond to an amino acid or a stop codon. Initiation of translation begins with the ribosome scanning the mRNA for the start codon. During elongation, the tRNAs bring the amino acids to the ribosome and transfer the amino acids onto the growing polypeptide chain. Termination occurs when the stop codon signals for the ribosome to dissociate, which releases the polypeptide. Since the carboxylic acid of an amino acid joins with the amine group of another amino acid to form a polypeptide, this gives proteins a directionality in which the first amino acid is at the N-terminus and the last amino acid in the chain is at the C-terminus. We have now gone through the basic processes of the central dogma of molecular biology. Thanks for watching.